Today's topic is neuromodulation as a new treatment for memory impairment or cognitive impairment in Alzheimer's disease, presented by Dr. Julian Keil today. And yeah, he's a former professor, so he's a doctor now. He was a professor at the University of Kiel. And still to this day, he is an associated researcher at the Clinical Neurotechnology Lab of the Charité in Berlin, and also an associated researcher at the Embodiment Cognition Group of Potsdam. And as well, he's the scientific director of the Neuron GmbH, which is the reason or the cause of today's presentation. His work in research mostly focused on perception, memory, and consciousness. And additionally, for working on neuron, he adapts or develops non-invasive and explanations, visual brain stimulations techniques to treat memory impairment and Alzheimer's disease of neuron. And also, like Professor Lee last week, he's a dedicated science communicator, wrote his own book, Biopsychology for Dummies, in English. And he has his own YouTube channel, which he more or less constantly uses for 5 years till now. And yeah, so now let's come back to the topic. Alzheimer's disease is probably one of the most known and worst diseases in the world. Everybody has at least heard once about it. And from what we know, we can't treat it how we would like to. And especially cognitive performance impairments won't adapt through pharmacological treatments. So we are constantly in search for new options, and this is where neuromodulation comes in as an option. And this is the point where I would like to hand over to you, Dr. Keil.
Excellent. Thank you very much for the kind introduction. I will just quickly share my screen. So I'm really happy to be here and very honored to be invited to give a lecture here in front of a dedicated student audience, because this I always feel that you're the next generation of neuroscientists. You're the next generation of researchers. So getting you interested in neuroscience, that's the core of my work, right? So today I want to talk a bit about neuromodulation as a new treatment for memory impairment in Alzheimer's disease. And in case you fall asleep at some point during my talk, or if you have to leave before, so here's the take-home message. So this is what I'm going to talk about today. So I want to convince you that we know a lot about the neuromechanisms of memory. So a lot of research in the past century actually has highlighted how we form memories, how we recall memories, and what processes are related to memory and how they are impaired in Alzheimer's disease. And I also want to convince you that neuromodulation can be a tool to modify these identified mechanisms of memory to then be a possible treatment for memory impairments, especially in Alzheimer's disease. Before we get started, please keep in mind, as Sebastian highlighted, I am a researcher in a startup. At Neuron, we are working on developing non-invasive brain stimulation methods to treat memory impairment, especially in Alzheimer's disease. So in the end, I will talk a bit about our latest research. So please keep in mind, take what I tell you about my own research with a grain of salt, because I'm very excited about this, but I have to be because that's my own work. So as always, if you talk to someone from industry, they have very strong interests in their own work. So keep a critical mind on this. All right, let's get started. Alzheimer's disease is one of the most problematic diseases that we're facing right now. Actually, people are, if you ask people about what they're afraid of getting sick in the future, most people highlight dementia as a stronger fear compared to, for example, cancer or other diseases. Currently, 55 million people suffer from different forms of dementia. And you are in a nice position that you are not affected yet by this, but as we age, the likelihood to suffer from any form of memory impairment or dementia vastly increases. So you can maybe find your age bracket on this trajectory. I'm a little older, so I'm close to 45 years point. So my trajectory points straight downwards. So for me, memory impairment is on the horizon and will be more likely as we as a society age, so the likelihood to the numbers of dementia patients will only increase. And importantly, this is not just due to aging, but also due to changes in our environment, but also within our body. For example, during menopause, hormonal changes actually increase the likelihood to develop dementia or develop Alzheimer's disease. So there is a very strong biological probability to develop dementia at some point. And currently, from these 55 million people suffering from dementia, most suffer from Alzheimer's disease. After Alzheimer's disease, the second most likely form is vascular dementia, so dementia is associated with heart conditions. The current idea of why dementia develops, or especially why Alzheimer's disease actually develops, is the so-called amyloid beta hypothesis. So this amyloid beta hypothesis has been around for quite a long time now, a few decades, and it is based on the finding that Alzheimer's disease is associated with a very marked neuropathology, namely mostly the accumulation of amyloid beta proteins in the brain. And this accumulation of amyloid beta proteins in the brain is neurotoxic, so it destroys neurons. And the leading hypothesis is that the death of neurons, so the atrophy of the brain, leads to the cognitive symptoms that we see in Alzheimer's disease. So if we look at the trajectory of the patients before they develop their first cognitive symptoms, we see that even before they show the first symptoms, there is strong changes in the way their brain looks like. So we have an accumulation of amyloid beta, with an accumulation of tau proteins, we have a loss of synaptic connections, we have a loss of neurons, and all this increases the risk of developing dementia. So the imbalance between the accumulation of amyloid beta proteins in the brain and the clearance of amyloid beta from the brain leads to the formation of these plaques, and these plaques can act as an initiator and accelerator of Alzheimer's disease. They are not a sufficient cause, so just having amyloid beta in the brain is not sufficient to develop Alzheimer's disease, but it can lead to the development of Alzheimer's disease. Still, most current pharmacological interventions are focused squarely on amyloid beta in the brain. So there's a line of new drugs developed to treat amyloid beta in the brain. They're called amyloid beta antibodies. They've been in the market in the EU since earlier this year, and they are very, very good at removing amyloid beta from the brain. So the idea of the development of these drugs was this amyloid beta hypothesis. So the idea was, okay, now we define Alzheimer's disease as this disease where amyloid beta accumulates in the brain. So let's develop a drug that removes amyloid beta from the brain, and then everything should be resolved. We remove amyloid beta from the brain, and the cognition should follow. People should be cognitively healthy again. And this research has been going on for at least 20 years. And unfortunately, the results are a bit mixed. So these antibodies that label amyloid beta to have it removed from the brain, they work very, very well to remove amyloid beta from the brain. So within the course of a year and a half after the beginning of the treatment, you can have an Alzheimer's disease brain completely free of amyloid beta. So you can get people to an amyloid beta negative state. However, this does not really translate to any improvements in their cognitive effects. So if we look at the decline of the cognitive abilities while people are in this treatment regime with these amyloid beta antibodies, there still is a large decline in the cognitive abilities. And with the treatment, the decline is slowed down by roughly three months. So it's a significant reduction in the decline of cognitive abilities, but it's not very meaningful. So you gain 3 to 6 months of of healthy cognition. And this comes at a price of severe side effects. For example, these amyloid beta antibodies lead to brain swelling. They can lead to hemorrhages in the brain. You can't take them if you have a heart condition. So there are several problems associated with these newly developed drugs, as is usual with new drugs on the market, and they might be refined in the future. Still, there is a need to develop therapies that work better for cognition. And here, neuromodulation can play a role. So instead of targeting the neuropathology of Alzheimer's disease, so instead of trying to remove amyloid beta from the brain, why not use a neuromodulation method, for example, brain stimulation, to directly activate the neural mechanisms of memory? So try a brain stimulation method to induce the memory-specific brain activity to directly improve cognition. And maybe this can also have indirect effects on the neuropathology as a second way. So the logic is different. So while pharmacological treatments directly target the known neuropathology of Alzheimer's disease, so they try to remove amyloid beta from the brain, neuromodulation approaches take a different route. They sidestep the neuropathology and try to directly influence the neural mechanisms of memory. Are there any questions so far, or did you follow the basic idea of the neuropathology of Alzheimer's disease?
So I would have one or two short questions. Depends on the length of your answer, I guess. The first one is, you mentioned that with the menopause, the risk of Alzheimer's disease increases, right? So I would guess that pharmacological or hormonal substitutional therapy would decrease this increase in risk, right?
Yeah, correct. Yeah.
Okay, perfect. Then, so it might be directed or linked to female hormones, or?
Yeah.
So has there been any research in treatments that are focusing on this pathway and not on mainly removing beta amyloid?
Yeah. So the hormone that is linked to this change in menopause is estrogen, and it is a very specific form of estrogen. I think it's called estrogen 2. And the amount, so the change in the metabolism of this estrogen 2 affects the likelihood to develop Alzheimer's disease. And there are hormone replacement therapies to slow down the decrease of estrogen level in the body. So also these are basic therapies for menopause to alleviate the physiological changes during menopause. The downside is that these treatments increase the risk of cancer. So they are very cancerogenic. So playing around with the hormone system also has severe side effects. But at least here we know, okay, it's estrogen, and modulating estrogen can dampen this effect of menopause.
Thank you. Would you like to have the second question right now, or would you like to
go ahead?
Okay, perfect. So one of your last points was that maybe the neuromodulation isn't only increasing cognitive performance, but also maybe affecting the physiology.
Yeah.
So, but there is a genetic defect or reason, I think, that there is more of the so-called bad beta amyloid produced. Mm-hmm. And is there anything like epigenetic connections that could say, okay, neuromodulation could affect this production? Because in the end, if this is the case, or the main reason why someone is developing Alzheimer's disease, neuromodulation wouldn't affect this aside from epigenetic effects, or?
Yeah, so the genetic effect, defect or genetic marker that you're talking about is the APOE4 gene. So it's a gene that is related to the production of amyloid beta, and carriers of this APOE4 gene variant have a higher likelihood to develop Alzheimer's disease due to having this accumulation of amyloid beta in the brain. And I don't think that neuromodulation can have any influence on this genetic pathway. However, I will come to this in the end, that neuromodulation could have an influence on the clearing mechanism. So I mentioned that the imbalance between the accumulation of amyloid beta and the clearing of amyloid beta from the brain leads to an accumulation of this amyloid beta. So if the clearing mechanism doesn't work properly, you get this accumulation of amyloid beta in the brain. And it might be possible to up-modulate the clearing mechanism so that even though you accumulate amyloid beta, you can also remove it from the brain. But I will come to this in the end.
Thank you very much.
Okay. All right. I don't see any other questions, so let's move on to a little side note on the basis of neuromodulation, because to understand neuromodulation, we need to have a short excursus or side path to the concept of neural oscillations. So you are psychology students, most of you, I would assume, and you have had your lecture in biological psychology, I hope. So this should be a little reminder of what you've learned in biological psychology. So you know that your brain is made up of different layers. So you have the gray matter and the white matter, and the gray matter itself mostly consists of 6 layers of neurons. And we have, for example, the layer 4, which contains a lot of pyramidal cells, which is the input layer of the neocortex. So that's the layer of your gray matter where the bottom-up signals arriving from your sensory system arrives. And these pyramidal cells, or these cells in your gray matter of your neocortex, they come in different variants. For example, you have these excitatory pyramidal cells and you have inhibitory interneurons. And both of them, and together, these excitatory and inhibitory cells process information that is coming from the outside, so bottom-up information, or also top-down information, and the information that is distributed throughout your brain. And the interaction of excitatory and inhibitory neurons create synchronized neural activity. So for example, an incoming bottom-up signal excites the excitatory pyramidal cells, so increases the firing of these pyramidal cells, and then the inhibitory interneurons, they pace these excitatory pyramidal cells so that all the pyramidal cells fire together in a rhythm. And then this synchronous firing, you might remember from Hebb's rule, cells that fire together wire together. So a bit longer formulation of Hebb's rule is that cells that are active at the same time, they form neural networks. So the influence of inhibitory interneurons on the excitatory pyramidal cells creates this synchrony in the firing of cells, which can then form neural networks to commonly perform information processing. So if we look at the spiking, so the action potentials of these pyramidal cells, we can see that they form more or less rhythmic, coherent activity. So a lot of these cells fire at the same time. And if we not look at the action potential of these cells, but record the field potential around these cells, so we record the summed activity of large groups of cells, then we can see neural oscillation. So we can see this rhythmic change in the polarity of the extracellular field. So we can see these rhythmic changes in the electrical activity in the brain. And these action potentials, they lead to postsynaptic potentials. And these postsynaptic potentials, they travel down the apical dendrites of the pyramidal cells. And the nice thing about these apical dendrites of the pyramidal cells is that they are in the neocortex mostly aligned in a parallel way. And the postsynaptic potentials in these parallel organized apical dendrites, they can actually sum up to such strong extracellular currents and local field potentials that you can measure this electrical activity outside of the brain, for example, with an EEG. And that's the basis of neural oscillations. So neural oscillations represent the summed activity of large groups of neurons that are active synchronously at the same time. And these neural oscillations are thought to represent the sort of language of neural processing, because we can see different aspects of neural oscillations are directly linked to information processing. So neural oscillations in different frequency bands, they allow the parallel information processing and transfer. So if we look at the time signal of a neural signal that we record through EEG, for example, if we look at so this is a time signal of activity of data recorded through EEG. So you have on the x-axis is time, on the y-axis is the amplitude. So the electrical activity recorded outside of the brain, it fluctuates, it changes over time. And if we decompose this time signal into different frequency bands, we can find the correlates of these different frequency bands with cognition. So for example, we can bandpass filter this time signal. So that means we pick out one frequency and then use a Hilbert transform to look at the amplitude and the phase of such a neural oscillation. This allows us to track how this specific frequency changes over time. We can also do a Fourier transform, which is a similar mathematical principle, but it not only allows us to look at one specific frequency, but it allows us to look at all the frequencies that are present in the signal, but we lose the time information in this case. And then there's a solution that combines the time and the frequency component, which is the so-called time frequency analysis. One method of this is the so-called wavelet analysis. And this allows us to track the amplitude across time of multiple different frequencies. And using this analysis, we can find out which different frequency bands are related to what kind of cognitive functions. And this allows us to identify parallel processing streams in the brain. And we can also find out how these different frequencies interact with each other. So for example, in the classical EEG that we record from the surface of the brain, we can identify neural oscillations between 1 hertz, so 1 cycle per second, up to hundreds and 50 cycles per second in the high gamma band. And these different frequency bands, for example, the delta and theta band, are mostly related to sleep or memory processes, while alpha and beta are related to attention and sensory processing, and the gamma band, the highest frequency band, is related to bottom-up stimulus processing or signal processing. And we can also find that the information transfer is instantiated in these different frequency bands. For example, there are nice papers showing that bottom-up stimulus, so incoming stimulus processing, is related to a gamma band, so high frequency oscillations, and the other direction, so top-down stimulus processing. So for example, the effect of attention on your visual processing is instantiated in low frequency bands, for example, in the alpha or theta band. Now, these different frequencies of neural oscillations are related to cognitive process. There have been a lot of papers showing the correlation between these different frequency bands and different cognitive processes. And now let's focus on memory and find out which are the frequency bands that are related to different memory processes. So if we take a step back and think about what is memory, how does memory work, we see that what is happening right now is that you are now observing a lecture and you're seeing a very specific information content. You're seeing my image, you're hearing my voice. So these are all different bits and pieces of information. Also, before you heard about the amyloid beta hypothesis, now you're hearing about neural oscillations. So these are different types of information. And these different bits and pieces of information that are stored in your short-term memory in separate neural networks. And at some point, these different neural networks need to be integrated into a stable long-term memory trace and into a stable long-term memory, so-called engram of the memory. And for this consolidation of these different neural networks into one coherent neural network, a very specific form of very high frequency oscillation is relevant. These are called sharp wave ripples. So sharp wave ripples are very, very short episodes of neural oscillations. They are roughly 100 milliseconds long in a frequency band of roughly 100 to 150 hertz. And they are related to the binding of these different neural networks into one coherent memory networks. And these sharp wave ripples are also related to the reactivation of neural networks that are in your brain. So once you have learned something and once something has been consolidated into an engram, then this engram can be reactivated by these sharp wave ripples. And these sharp wave ripples are controlled by low frequency oscillations. So the high frequency sharp wave ripples are related to binding of these different neurons into neural networks, and the timing is controlled by low frequency oscillations. This has been the basis of a very, very famous paper by Jensen, by Lisman and Idiart from the 90s, who argued that the low frequency oscillations creates windows of opportunity for the activation of these single neural networks in high frequency oscillations. And here Lisman and Idiart argued that you can fit roughly 7 cycles of a high frequency oscillation in one window of a low frequency oscillation. So they argued that the fact that you can memorize roughly 7 plus minus 2 items in your working memory is related to this nesting of high frequency oscillations to the up phase, to the active phase of a low frequency oscillations. So these networks of synchronously active neurons, they store the information. They are active within the cycles of a sharp wave ripple, and the timing of the activation of these sharp wave ripples and the timing of the activation of these networks is controlled by low frequency oscillations. This was a theoretical model back in the 90s, and then subsequently people have shown that this is actually the case. So if we look at recordings from the hippocampus, so direct electrical recordings from the hippocampus, you can see this combination of low frequency oscillations, and then at specific phases of the low frequency oscillation, you see these high frequency oscillations riding on top of these low frequency oscillations. So there's a nesting of low frequency and high frequency oscillations. Low frequency oscillations, they control the timing. High frequency oscillations, they control the binding of these different forms of information. And using animal research, we can actually track the activation of these neural networks. For example, in classical mouse learning experiments, you have mice learning their way across a maze. So they create spatial memory. And you probably have heard of place cells and grid cells in the hippocampus and the surrounding neural tissue. And so, for example, every part of this maze or of this path is represented in a different place cell. So each of these lines here is one cell firing, and depending on where the mouse is, different place cells fire. And then during memory consolidation, these sharp wave ripples, they reactivate these place cells and consolidate these different bits and pieces of information into one coherent memory. So again, the critical part here is the high frequency oscillations, the sharp wave ripple, they lead to the binding of this information, and the timing of the activation is controlled by low frequency oscillations. Again, so far so good. Any questions so far, or did you get the basic idea of different frequency bands and the importance of nested information for controlling the activity of these different frequency bands?
Maybe I have a question.
Yes, please.
Stupid questions, but I don't recall this wrong from at school. But aren't there also sharp waves regarding epilepsy? Epilepsy?
There are similar looking waveforms during epilepsy. So these are called the ictal spikes. So during epilepsy, you have a strong synchronous discharge of activity throughout the brain. This is the hallmark of an epileptic seizure, right? So you have a part of your brain that is malformed or has a malactive function, and this creates a spreading activation throughout the entire nervous system, and this creates this synchronous sharp waves of activity throughout the brain. And this can look very similar to the sharp wave ripple activity. The core difference is the location of these ripple oscillations. So in an epileptic seizure, you have them all over the brain, synchronously throughout the brain. These memory-related sharp wave ripples, they are localized to memory-related areas, for example, the hippocampus. And they also differ in the number of cycles. So usually the ictal spikes have one large deflection, and that's it, while these sharp wave ripples have multiple cycles of an oscillation. And actually, these multiple cycles represent the reactivation of these multiple neural networks. So you're absolutely right. It can look similar, but it has different, well, epilepsy doesn't have a function, but it has different outcomes.
Thank you very much.
All right, so now we know there are neural oscillations in the brain, and these neural oscillations are related to memory and memory processes. So basically, we now know how memory works. So we know there's different forms of memory. There's episodic memory, there's declarative memory, there's motor memory, but all these different forms of memory are related to the binding of information to neural networks. And actually, there are people who argue that all memory is just sequence learning. So for example, Georges Boujaki, one of the most prominent memory and sharp wave ripple and neural oscillation researchers worldwide, his main argument is that all memory is just sequence learning. You can break down any type of memory to what is this path learning in animals, so learning a sequence of events. That's his main argument, and maybe we can have different views on that, but that's his main argument. And so we basically know how memory works. And now the next question is, can we directly modulate these different neural oscillations to improve memory? And here the next concept becomes important, which is entrainment. So entrainment allows modulating neural oscillations with sensory or electrical stimulation. So if I give you a stimulus, so if I show you a picture, so let's see, I don't have a picture with me. I show you a picture of the brain. So this picture of the brain evokes a synchronous activity in your visual cortex. So the stimulation can reset the ongoing activity in your brain. And if I don't show you this picture just once, but I show it to you repeatedly, this repeated series of visual evoked potentials can entrain neural activity to evoke cortical activity in the same frequency as the stimulation. So this rhythmically repeated stimulation can induce neural oscillations in the stimulation frequency. And so if we use this stimulation in the same phase as an ongoing oscillation, so for example, we have this theta or alpha oscillations, very strong oscillations in the brain. And if we use this stimulation in the same timing, in the same phase with these ongoing oscillations, we can actually increase or decrease the amplitude of these ongoing oscillations. And if we have strong enough stimuli, that's what we see up here. So if we have very, very strong stimuli, then we can actually artificially create oscillations in the brain that are not related to any ongoing processes. So we can, using entrainment, we can either modulate stuff that is ongoing, so stuff that is already there, so ongoing oscillations that are already there, or if we have strong enough stimuli, we can actually create artificial oscillations just by rhythmic repeated stimulation. And usually this takes a while to build up. So if we look at the EEG signal or during this rhythmic sensory stimulation, you can see this build up of the response of the brain, and at some point you can create these very strong oscillations in the brain. And this entrainment of oscillations in the brain, that is the basis of the neuromodulation that I want to focus on today. Because we now know that different neural oscillations are relevant for memory. So a little repetition. So for example, we know that slow oscillations in the neocortex, slightly faster oscillations in the thalamus, and these very fast oscillations in the hippocampus are related to memory processing. And they form a system, an interactive system that controls memory consolidation and memory recall. So these different oscillations are relevant for different memory processes. And now we can use sensory stimulation or electrical stimulation to induce these oscillations by rhythmically stimulating the brain. A little side note before I go into the details of these different entrainment methods. These are all non-invasive stimulation methods, but there are also invasive stimulation methods. For example, deep brain stimulation, where you implant an electrode in the brain to act as a stimulus. And there are also neuromodulation methods that are not related to an entrainment, such as, for example, vagus nerve stimulation. So the vagus nerve is one of these cranial nerves that control the interaction between brain and body. And for example, you can stimulate the vagus nerve by stimulating a part of your ear, and that way you can modulate activity in subcortical brain structures such as the thalamus and associated areas. Deep brain stimulation also doesn't necessarily use entrainment, but changes the excitability of large-scale neural networks. So these are different stimulation methods or neuromodulation methods that are not related to entrainment, and I won't focus on them right now, but I will focus on non-invasive brain stimulation methods that use entrainment as a core mechanism. So for example, we can use rhythmic magnetic pulses delivered with transcranial magnetic stimulation to increase rhythmic pulses in the brain. This is called repetitive transcranial magnetic stimulation. And this is a very precise way to stimulate very, very small circumscribed areas of the neocortex. So for example, we can stimulate the motor cortex to differently modulate different parts of your fingers. So you can, with a stimulation of your motor cortex, you can activate single fingers and control the movement of single fingers of your participant. So this is a very, very precise way of stimulating the neocortex. Transcranial direct current stimulation or transcranial alternating current stimulation is also a stimulation method based on electrical stimulation. There you place different electrodes on different parts of your brain, and then a current flows between these electrodes. And with this stimulation, you can actually also reach deeper areas of the brain to induce oscillations in the brain. And then there is sensory stimulation, which I will focus on in the end, where you can use, for example, visual stimuli. Okay, you can use auditory or tactile stimuli to induce these sensory evoked potentials, which can then lead to oscillations in the brain. So you can use this to stimulate the brain via the sensory pathways. So I first want to focus on the use of TDCS or TACS to induce slow oscillations in the brain to improve memory. There has been a very prominent paper by Linda Marshall from 2006, who used transcranial direct current stimulation of the forehead to induce slow oscillations during sleep. So again, if we think back of the memory processes and the role of different oscillations in memory processes, these researchers, they tried to induce slow oscillations which are related to the control of the timing of high frequency oscillations. So in this experiment, participants were asked to learn a list of words. So participants were asked to memorize a list of words, and they were also asked to learn a motor sequence task. And then the participants were allowed to sleep. And during sleep, they were stimulated on their forehead with transcranial direct stimulation, rhythmic transcranial direct stimulation at a frequency of 1 Hertz. So you can see this peak here at 1 Hertz. So with this stimulation, they increase the power of these delta oscillations, so these 1 Hertz oscillations. And then after sleeping, people were asked to recall the words they had learned, and the researchers showed that the participants who were stimulated with the transcranial direct current stimulation had a better recall than those participants who received a sham stimulation. So this is an experiment showing that amplifying these slow oscillations during sleep, where you consolidate your memory to one coherent engram, can actually improve memory. And this not only works with transcranial direct current stimulation, but it also works with sensory stimulation. So in this paper by No from 2013, it was essentially the same experimental setup. It's also the same research group. Again, participants were asked to learn a word list, and then during sleep, these researchers used auditory sensory stimulation to stimulate at the excitatory phase of delta oscillations. So using EEG, the researchers detected the phase of these ongoing delta oscillations and then used rhythmic stimulation to increase the amplitude of these slow oscillations. So again, there was a stimulation group and a sham group. And again, these researchers show that the memory retention or memory recall was increased with this slow frequency sensory auditory stimulation compared to a sham stimulation. And they could also show that with the actual real stimulation, they could increase these low frequency oscillations. And they also had a sham a control condition where they changed the timing of the stimulation to actually interfere with these slow oscillations, but this didn't have any effect. So this didn't impair memory, but increasing these low frequency oscillation actually improved memory in this experiment. So this is evidence or indication that amplifying these slow oscillations that are relevant for the timing of neural activity can improve memory, can boost memory consolidation. But you remember, slow oscillations isn't anything. Slow oscillations, that's controlling the timing, but the actual formation of neural networks, that's related to these high frequency sharp wave ripples. So what the question is now, what happens if we control sharp wave ripples? And interestingly, controlling sharp wave ripples allows to modulate memory in both directions. So this is research that has been done in mice, because the control of these sharp wave ripples so far only worked with very invasive methods. For example, electrical stimulation or optogenetic stimulation, with which you can control the activity of single neurons. And using these methods, you can actually control these sharp wave ripples. So for example, in this paper by Zhang, they used an optogenetic stimulation to block the occurrence of these sharp wave ripples. So in this experiment, animals were asked, or were not asked, animals had to memorize a way through a maze. So they had to memorize a path. And then after these mice had learned the path, the occurrence of the sharp wave ripples was blocked. So for example, here in the blue line, you can see, so these mice learned their way through the path. And then after half of the experiment, the occurrence of the sharp wave ripples was blocked. And these mice actually forgot. They couldn't remember the way through the maze anymore. So if you block sharp wave ripples, you block the recall of memory. And it also works the other way around. So if we increase the number of sharp wave ripples during sleep, so during memory consolidation, this actually improves learning in mice. So in this paper by Oliva, the authors increased the number of sharp wave ripples. So they artificially triggered sharp wave ripples using optogenetic stimulation. And this this improved learning in these mice. And in this experiment by Fernandez Ruiz, they prolonged the sharp wave ripples, and this increased learning. So these mice with the longer sharp wave ripples, they were faster to learn, to find their way, to learn the strategy to find the way through the maze. So blocking sharp wave ripples actually impairs memory, and boosting sharp wave ripples improves memory. So there's this different modulation of switching memory on and switching memory off by controlling sharp wave ripples. So now we have the tools to actually modulate memory by either controlling slow wave oscillations or controlling sharp wave ripples. Unfortunately, so far, sharp wave ripple generation or sharp wave ripple induction isn't really feasible in humans, because these methods that are used so far are involve either a genetic manipulation for optogenetic stimulation, or use invasive electrical stimulation, which isn't really feasible in long term in humans. And now we come to the part where I would ask you to be a bit more critical, because now we come to my own research, because now this is what I've been working on developing a method to induce sharp wave ripples in the brain of humans, so to safely induce sharp wave ripples in humans. And for this, we have developed a high frequency visual stimulation. So again, we use entrainment, we use sensory entrainment to induce oscillations in the brain. But we are using a very specific way of entrainment, a spatiotemporal organized sensory entrainment that is inducing a lot of parallel oscillations in different parts of the visual system. And our core argument is that using this parallel activation of the visual system is with this method, we can induce high frequency oscillations in the brain, and these high frequency oscillations in the brain then propagate down to the hippocampus to induce these memory related sharp wave ripples. So this is our basic hypothesis of what we are working on. And we have conducted a first experiment in mice, where we asked whether we can actually use this ultra high frequency visual stimulation to get into the brain. So can we pass the limitations of the retina? Can we actually process these high frequency signals? And can we induce high frequency oscillations in the visual cortex of these mice? And long story short, yes, we can. So what you can see here on this plot is the time course and the number of channels. So in this experiment, we had mice with implanted neuropixel probes. So neuropixel probes are longitudinal electrodes with a lot of different electrode contacts. So overall 384 electrode contacts. And along these different electrode contacts, we can record neural activity. And what we see here is this so-called steady state visual evoked potential along the different parts of the visual cortex. So we can see that using our high frequency stimulation, we can induce these neural oscillations in the brain. And you can actually, if you look at a single channel, you can actually see these, you can very nicely see this entrainment of the visual system using our stimulus. And importantly for us, what you can see down here is that if you look at the frequency scale here, we increase the power of these high frequency oscillations using our stimulation. So the first part of our hypothesis seems to work. So we can use a sensory stimulation, a rhythmic sensory stimulation, to induce an entrainment, to entrain the visual cortex to increase the power of high frequency oscillations, at least in mice. So that was the first step of our development process. And then we went to epilepsy patients, because epilepsy patients, so there is a small group of epilepsy patients who are treatment resistant. So they have seizures despite getting anticonvulsive medication. And to find out where the cause of these epileptic seizures is, some of these patients get electrodes implanted into their brain for diagnostic purposes. And some of these patients have these electrodes implanted in the hippocampus. And we are very lucky that we are able to record, to conduct experiments in this very selected patient group. And there, in these human patients with the electrodes implanted in the hippocampus, we can test whether our stimulation actually induces sharp wave ripples in the brain. So we had, there's a typo. It was actually 8 patients. So 8 patients with these implanted electrodes in the brain, with overall 42 electrodes sitting in the hippocampus. And then with these patients, we used our high-frequency visual stimulation, first of all, to find out whether it's safe for use in humans, and second of all, to see whether we can induce sharp wave ripples in the brain. Long story short, yes, we can. So using this high-frequency visual stimulation, we can actually induce sharp wave ripples. So here you can see an average across these sharp wave ripples of all these 8 participants. You can see, again, this slow wave and this high frequency oscillation riding on top of this slow wave activity in the brain. And during the stimulation, we have more sharp wave ripples than during baseline recording before or after the stimulation. So we're quite confident that our stimulation is able to induce sharp wave ripples in the brain, so increase the number of sharp wave ripples quite significantly. So this is a paper that it's ready. I hope to be able to submit it tomorrow, where we show that using our high frequency visual stimulation, we can increase the number of sharp wave ripples. And now the crucial question is, why? What's the point? Well, yeah, you can increase the number of sharp wave ripples. What's the use? So we conducted an experiment in psychology students to see whether we can increase the memory of psychology students. So we had 17 healthy participants. And again, we used a word learning task. So very, very similar to the work of Linda Marshall that I've shown you before. So the students were asked to memorize a list of words. And then afterwards, they were stimulated for 5 minutes. And then they were asked to recall the list of words. And then we repeated the same thing. So we had a second run of encoding, a second run of stimulation, and a second run of recall. And then we measured the learning rate. So how much does learning improve from the first to the second stimulation? And we had a fully within participants design. So the same participants either first received the high frequency visual stimulation or a controlled stimulation, or the other way around, started with a low frequency with a sham condition or with the actual stimulation condition. So we have fully parallelized within participants design. And in this experiment, we saw that the learning rate, so the improvement from one learning to the next learning, was higher during the stimulation condition than during the sham condition. So at least in psychology students, 5 minutes of high frequency visual stimulation appears to improve the consolidation. So remember, there's a stimulation during the rest period after learning. So apparently supports the memory consolidation to improve memory recall in the students. So this is now for us the starting point to develop a treatment for Alzheimer's disease patients. So for us, this is now what we're working on, trying to translate this fundamental research into applied research, where we develop a medical device, a medical product as a potential therapy for Alzheimer's disease patients. And interestingly, so Sebastian, you asked this before, okay, this is cognition, this is memory. Might there also be effects on the neuropathology of Alzheimer's disease or memory impairment? And I do have a hypothesis, or quite actually 3 hypotheses on that. For example, sharp wave ripples are related to the formation of synaptic connections. So you might have heard about long-term potentiation, right? Long-term potentiation, the fundamental basis of synaptic connections and forming neural networks. And sharp wave ripples are one of the processes that induce long-term potentiation. So long-term potentiation is increased during phases of high frequency neural activity. And so sharp wave ripples are high frequency activity. So during sharp wave ripples, long-term potentiation actually happens, and this can strengthen synaptic contacts. So increase in number of sharp wave ripples could counteract the loss of synaptic connections in Alzheimer's disease. Similarly, there are now a few papers showing that sharp wave ripples are related to the activation of the glymphatic system. The glymphatic system is a very interesting system that has been discovered in 2012, researchers discovered that there is a waste removal system in the brain, similar to the lymph drainage system in the body. And this lymphatic system is a system that removes toxins and waste products from the brain. And this system is activated during sharp wave ripples. So if we increase the number of sharp wave ripples, we could increase the clearance of waste products from the brain. And this points back to what I said in the beginning, this balance between the accumulation of waste products and the clearing of waste products. So if we ramp up the clearing of waste products, we could also have a beneficial effect on the neuropathology. And then finally, and this gets a bit more speculative, sharp wave ripples are also related to the activation of glial cells. And you might know that glial cells are related to the myelination of the axons of neurons, so the oligodendrocytes in the central nervous system are glial cells that insulate the axons of pyramidal cells. And in Alzheimer's disease, these oligodendrocytes are actually impaired. And we know that sharp wave ripples are indirectly related to the maintenance of these oligodendrocytes. So again, there might be a possibility to enhance the well-being or structural integrity of these oligodendrocytes by boosting the number of sharp wave ripples. So very, very speculative, but increasing the number of sharp wave ripples also could be a potential treatment for the neuropathology of Alzheimer's disease. All right, so let's summarize. The likelihood to suffer from memory impairments, especially Alzheimer's disease or dementia, increases with age. And since our societies are aging, we are all getting older, the risk of developing dementia is rising. And so the question is, how can we counter this rise in dementia? The current hypotheses on the development of Alzheimer's disease, namely the amyloid beta hypothesis, is not really sufficient to explain dementia. It is related, but it's not a perfect correlation. And also this just removing amyloid beta from the brain isn't a cure for Alzheimer's disease. So it slows down the cognitive decline, but it doesn't cure it. It doesn't stop it. So there's a need for other therapies. Neuromodulation is a way to target the known mechanisms of memory. So we now know that the combination, the nesting of low frequency and high frequency oscillations, especially delta oscillations during sleep and high frequency oscillations during memory consolidation, also related to sleep, are the mechanisms of memory consolidation and memory recall. Using neuromodulations, we could directly target these neural oscillations to amplify them and to boost memory performance and cognition with these stimulations. And it might also be possible to then develop treatments for Alzheimer's disease, so shifting the focus from just the neuropathology to the mechanisms of memory allows developing therapy options to directly target cognition and memory impairment in Alzheimer's disease. So thank you for your attention. And as Sebastian mentioned in the beginning, I wrote a book, Biological Psychology für Dummies. So if you want a very concise and very hopefully easy to understand summary of biological psychology, also a bit related to memory, you can read it up in my book. And now I'm curious to hear your thoughts on the use of neuromodulation as a potential therapy for memory impairment.
First of all, I would like to thank you for this very nice presentation, very nice insights. As Dr. Hill mentioned, if there are any questions right now, you feel free to ask them. I still have one or two, but I would like to give you the chance first. So therefore, I'll wait. So if anyone would like to ask his or her question, just feel free.
Yes, Azuka.
Yes, thank you very much for the talk. First of all, it was very interesting. So I actually have multiple questions, but I'll try to put it in one concise question. So about the data you showed from your own research,
Yeah.
The differences in the in the short wave ripples in the clinical sample. Yes. So my first point would be, there is like a considerable, or I would assume there's a considerable difference between individuals how well the
Yeah.
enhancement of shortwave ripples
Yeah.
work. And linked to that, if you go to the student sample, there's also very big differences in the enhancement of the learning rate, and for some people even a decline in the learning rate. Is that somehow connected to the effectiveness of inducing more shortwave ripples in the participants, that for those where it works better, you see like significantly better results in learning rates as well? And if so, do you have like any hypothesis on what may cause the individual differences in effectiveness of shortwave ripples, or even the induction of shortwave ripples in the first place?
Excellent question. So the short answer is no. The long answer is yes, there is considerable difference in the ability to induce sharp wave ripples. So if we go to this figure, you see there's a very, very large spread. And this is, remember, these are log transformed counts data. So there is a lot of participants who show overall very few sharp wave ripples and also show a very small improvement in the number of sharp wave ripples. And then there is a few that show a very large difference. So it's a Poisson distribution here. One problem with this kind of research is that we can't control where these electrodes are in humans. So these are electrodes that are placed for diagnostic purposes, and we hope that they are in the hippocampus or close to the hippocampus. So some of these electrode contacts might not be in ideal places to actually record sharp wave ripples. So if you can't record a lot of sharp wave ripples, there's also a little you can change. So placement of electrodes definitely plays a role. Second of all, these are epilepsy patients who are on different levels of medication. So these are patients who are recorded during monitoring for epileptic seizures. So some of them are still on anti-epileptic medications, some are not. And these different levels of medication also might influence the sharp wave ripple generation. Unfortunately, the experiment where we directly link the number of sharp wave ripples to the memory performance is currently only running. So I had the first patient on Tuesday. So Sebastian, when we were calling, I was actually on the way back from Bielefeld, where we had the first patient where we combined a memory task with the induction of sharp wave ripples. So unfortunately, I can't really tell you how the number of induced sharp wave ripples is related to memory performance. There is, however, a very, very nice paper by Itzhak Norman on the relationship between the number of sharp wave ripples and memory performance. And he showed a very tight correlation between the modulation of the number of sharp wave ripples and memory performance. So in this experiment, give me a second. I think I have a slide on that. Yeah, here it's the left graph here. So in this experiment, participants were asked to view videos, and they were asked to memorize the content. No, not videos, pictures. So they were asked to memorize pictures, and then they were asked to recall the pictures after a resting phase. And those participants who had a strong modulation of the number of sharp wave ripples between items that they could remember and items that they forgot. So roughly translated is patients who could increase the number of sharp wave ripples during learning and during memory recall, they also had the highest number of items recalled. So there's a direct correlation between the number of sharp wave ripples and the amount of information you can recall. And associated with this paper, so Norman et al. 2019, is a super impressive video where they have the audio recording of these patients recalling what they learned. And as a sound trace, you have the occurrence of the sharp wave ripples. And it's super impressive that you can hear the sharp wave ripples coming up, and then shortly afterwards, the participants verbalize their memory content. So this is a very nice illustration that sharp wave ripples and the occurrence of sharp wave ripples is related to this reactivation of this neural network. So there is a quite impressive correlation between the number of sharp wave ripples and actually memory recall. Does this answer your question?
Yes, thank you very much.
Okay. Fedor.
Yes, thank you very much first for the very interesting talk. I have just a short and common question to the Alzheimer's disease at all. So you have mentioned several times that the removing of these amyloid plaques did not bring any improvement to the treatment. So can we conclude out of this that these amyloid plaques are just a symptom, but not the reason for the disease, that there are any reasons?
You basically are at the heart of an ongoing discussion about what is Alzheimer's disease actually. So the amyloid beta hypothesis that I mentioned here, it has dominated the research field for the past 20, 30 years. Actually longer. It was established back in the 80s, so close to 40 years already. And this amyloid beta hypothesis was the first hypothesis on the neuropathology of Alzheimer's disease. And following this discovery that there is a lot of amyloid beta plaques in the brain of Alzheimer's disease patients, a lot of research was focused solely on what's the role of amyloid beta in the brain. However, the big problem is that we all have amyloid beta in the brain. As you age, amyloid beta accumulates in the brain. But this doesn't necessarily mean that you actually develop Alzheimer's disease. There has been a very prominent study in a group of nuns who, as you might know, nuns, they live a very stress-free life. So they have a very structured life centered around work and prayer. And these nuns, they got very old. And after they died, people evaluated or examined their brains, and they found that these nuns, these very old nuns, had a lot of amyloid beta in the brain, but none of them actually developed dementia. So they're just having amyloid beta plaques in your brain doesn't mean you develop dementia. So it's not a sufficient cause of amyloid beta in the brain. And there is now researchers actually calling this amyloid beta hypothesis a dead-end street and a wrong turn in for research. So I wouldn't necessarily go that far, because I think amyloid beta does play a role in the development of Alzheimer's disease. But as I said, it's likely just a puzzle piece in this very complicated picture of different pathologies that amplify and interact with genetic markers, interact with stress, that can then increase the risk of developing dementia. So long story short, I would say amyloid beta can't be the only thing, because it just doesn't work well enough.
Yes, thank you very much.
Clara.
Yes, hi. Thank you very much for your talk. I've got a short question. It's just that your research, or brain stimulation, is mostly focusing on treating symptoms, right? So you can't really improve, or you can't find a cure by stimulating the brain.
Yeah, that's I was kind of hinting with this at this point with
the last
with this
slide.
Yeah, with the last slide. So you are absolutely right. So the main focus right now is improving the symptoms of the Alzheimer's disease. There we have the strongest hypothesis. So there is research already showing that modulating neural oscillations can improve memory, can improve cognition. So that is our strongest hypothesis that we're working on right now. These links to the neuropathology, they are theoretical right now. We're right now running an animal study with Alzheimer's disease mice, where we use the same high frequency stimulation that we've used before. So using this same stimulation that we used here in Alzheimer's disease mice. And there we want to see, do we see any change in the neuropathology associated with this brain stimulation? There is another project working on a visual stimulation in the brain. It's developed at the MIT in Boston. They are using a 40 hertz visual stimulation, and they claim that this 40 hertz visual stimulation activates microglial cells to modulate neural activity and to improve the neuropathology in the brain. So there might be also links to the neuropathology, but this is I would say, more speculative than this very well-established link to cognition. And since there are therapies for removing amyloid beta from the brain, it also might be less relevant, but that is still to be found.
Thank you.
All right, Friedrich, I see.
So, hello, can you hear me?
Yes.
Okay, and then also thank you for this great presentation. I got a question concerning like this two mechanisms. As far as I understood that they're like this slow waves and also these short SWR, short ripples.
Sharp wave ripples, yeah.
Sharp wave ripples. And as far as I understood, making both of them stronger will enhance memory.
Yeah.
And I would be like interested, do we know what's the difference? So are there difference in effects if we either make the slower one stronger or the sharp wave ripples?
Excellent questions. Unfortunately, I don't know of any study that has looked at both. And this is a bit related to the way our research system is set up. So right now, people looking at sharp wave ripples are animal researchers. So for example, we are collaborating with the German Center for Neurodegenerative Diseases here in Berlin. We have brilliant colleagues who are experts in analyzing sharp wave ripples in brain slices or in mice or in rats. But these are neurobiologists and neuroscientists that are not really interested in, for example, humans or Alzheimer's disease or something like that. And then on the other hand, you have people like me who are interested in neuromodulation and neurostimulation. And these are people who are using humans. And well, in humans so far, it wasn't possible to induce sharp wave ripples. So people focused on what was possible, namely influencing slow oscillations. So you have these different fields of research who don't really talk to each other and don't really interact a lot. So there, as far as I know, there isn't any study that is looking at both. And this is also complicated by the fact that combining both, so low frequency and high frequency oscillation, requires very, very precise timing of both methods. So there is a paper from which I got this figure actually by Simon Hanselmeier, excellent review on entrainment and neuromodulation. And one of the core arguments of this paper is that the timing matters a lot. So the timing of stimulation matters a lot. And so it's rather difficult to get the timing right for slow oscillations. So as, for example, people have done in this paper, so you have to detect the slow oscillations and then have very precise timing of the stimulation of these low frequency oscillations. On the other hand, the timing of these high frequency stimulations might not be as important because you can just vastly increase the number of these sharp wave ripples, and this already has an improvement of memory. But what you mentioned is actually the crucial question. So what is the effect that is working better? What has the better outcome? Should we focus on low frequency oscillations? Should we focus on high frequency oscillations? Should we focus on both? That is actually something that we're still working on, and it is unfortunately still unknown, but might be an interesting research topic.
Well, yeah, thank you.
Or maybe you have a different idea.
Not yet.
Okay.
But yeah, really fascinating.
Yeah.
So I think right now there aren't any more questions from the chat. So I would like to ask mine, if it's fine with you.
Go ahead.
The first one is, so you're basically using a steady state response, right?
Yeah.
First of all, first question, you've used it on epileptic patients. And I think one of the main criteria to not participate in a steady state response study is having epileptic seizures. Wasn't there any problem, or was it due to the medication, or how were you allowed to do it?
So it was a very long discussion with the ethics committee. Yes. So the core thing is that these epileptic patients that we're using this on are you are in a hospital. So they are in the epilepsy clinic because they want to have seizures. So they are there for diagnostic purposes. They want to have seizures so that that the doctors can find out where the source of the seizure is coming from. So they are there to have seizures, and they are also very well supervised. So if a seizure happens, there's a medical team right on site. So that's why we thought, okay, there's little risk associated with actually inducing seizures. There was one reason. The second reason is that the visual stimulation frequency that induces seizures is actually rather low, so mostly between 15 and 30 hertz. So it's actually it's a frequency that you would observe with a stroboscope in a disco, for example, or in a club. That's the frequency that mostly induces these photosensitive epilepsy seizures. And we are in a frequency range far above this. So for example, we're using 120 or 140 hertz stimulation. So this is way above this frequency range that could potentially induce seizures. And then finally, luckily for us, in these patients that we recorded, we didn't observe any seizures. So apparently our stimulation is safe to use, even in vulnerable patients. But yes, that was a very grave concern that we had.
Thank you. And second, so I've known about entrainment, and currently I'm working with the steady state response for my first study. But I was thinking about if the steady state signals are evoking or mimicry of oscillations, evoking oscillations in patients, wouldn't that mean our results are getting confounded due to certain experiments? Because if I induce better oscillations in people, their attention might increase, and I am doing an intentional task right now. So I can say my results are mainly to my suggestion or my hypothesis. It might be due to the effect of oscillations, or?
Yeah, and that is an excellent question. And you're again pointing directly at one of the cores of this ongoing discussion about what is the role of neural oscillation. And let me quickly, I'm going to look for one paper. Give me a second. I stopped sharing my screen. Let me share my screen again. What was it? So give me a second. So there's an so there's a paper on gamma oscillations so sensory evoked gamma oscillations and endogenous gamma oscillations, they occur at the same time and they don't necessarily interact. Oh my God, I can't remember the name. Who was it? Here, this paper by Dücker. Here, Katharina Dücker. So they used gamma sensory entrainment and also used these. Oh, sorry, you can't. I forgot to show.
Yeah, I just wanted to mention it, but yeah, thank you.
Okay, sorry. So I was distracted. So this paper by Katharina Dücker. So they used either sensory entrainment or these looming grating stimuli that are known to induce gamma responses in the brain. So two different ways of inducing gamma band response in the brain: entrainment or looming moving stimuli. And the core argument of this paper is that both things happen at the same time simultaneously, but they don't interact. So you can induce or you can entrain a gamma band steady state visual evoked potential in any frequency you like, but this doesn't interact with the ongoing gamma band oscillation. So it's two different things, and this goes basically to the question of: are we creating artificial oscillations with entrainment, or are we capturing or hijacking an endogenous oscillation? And this is an ongoing discussion. So there are people who are favoring the artificial stimulation argument, as Katharina Dücker and Ole Jensen. And there are other people who are favoring the capturing argument. For example, as Simon Hanselmeier does, who argues that if you are in the right frequency band and the right phase and you have the right time, you can actually capture an endogenous oscillation. The complicated thing is that there is evidence for both. So there is, for example, a very nice paper by Katharina Bauer, who showed that, which one was it? Who used a visual stimulus to, no, an auditory stimulus to modulate visual cortex oscillations. So she used an auditory system to entrain the visual cortex in a certain oscillation, and then presented stimuli at different phases of this entrained oscillation. And this actually had an effect on stimulus perception. So depending on this entrainment, the signal detection differed. So with near threshold visual stimuli, and your ability to detect these visual stimuli changed according to the phase of this entrained oscillation. So there is evidence that it is possible to capture or modulate endogenous or attention-related oscillations, but at the same time, there's evidence for that these different oscillations can occur at the same time. So it is still an ongoing debate, and I'm actually leaning more towards this being an artificial signal, but I haven't found really conclusive evidence for either one of those.
Thank you. I think basically this, like every time in science, where
Yeah.
at least two options are possible. Yeah, by looking at the time, and first of all, I will look at the chat. Are there any questions? No. It is 9:30, so we have about 90 minutes. We would have 4 questions that were submitted, but weren't answered during your talk. For those who would like to stay, I think we could ask them right here, so that you, Dr. Keil, could answer them.
Yeah.
So I would post them in the chat and then also read them out one after another. So give me a second. But if you have any questions or any questions come up in your mind, just feel free to interrupt me. It's totally good. So first one is in the chat, and I also will read out. What are the main technical or ethical limitations in translating oscillation targeting neuromodulation from animal models to human clinical practice?
So the main technical difference or main technical problem is that in animals, we can be very precise in where we stimulate. So we can place, for example, stimulation electrodes wherever we want. So there are some limitations for ethical concerns, but overall, you're pretty much free to do any type of stimulation anywhere in the brain of a mouse or of a rat. In humans, invasive electrical stimulation is only permissible for therapeutic reasons. So for example, in the ECOG patients, we could theoretically also stimulate through the implanted electrodes, but we can't control where they are precisely. So it's not up to us to place an electrode. Similar for deep brain stimulation, for example, deep brain stimulation in Parkinson's disease, the electrode placement is guided purely by therapeutic reasons. There's also deep brain stimulation for depression, and also now is being tested for memory impairment. And there again, the stimulation placement is purely directed for therapeutic reasons. So we don't really have the freedom to test different stimulation areas to find what is happening where and what are the different effects. Second of all, some methods aren't suitable for humans. So for example, optogenetic stimulation requires the implantation of photosensitive, genetically manipulated photosensitive ion channels into nerve cells. You can't do this in humans. There's no way an ethics committee would allow genetic manipulation in humans. So this is a method that is just not feasible for humans. And I think this is good. This is a good reason to do this, because I think we have to be very, very careful with permanent stimulations in humans outside of a very well-known and very precise circumscribed therapeutic reason, because the chance of doing something wrong is so big. And even if you think about deep brain stimulation in humans or patients undergoing Parkinson's disease treatment, the placement of these electrodes is a very, very sensitive thing. We now know that it's the nucleus subthalamicus that needs to be stimulated. Placing these electrodes in this nucleus subthalamicus only works while patients are awake. So you have to monitor that you don't harm and you don't injure the brain of these patients during electrode implantation. And then you still have the problem that the hardware deteriorates over time. So the efficacy of deep brain stimulation declines with increasing time. So you have to update the stimulation, you have to change the stimulation, you have to change the electrodes after some time. So actually, I would say for humans, invasive stimulation is the last resort. And if we can go non-invasive, I think that's a lot more feasible in humans.
Thank you. And I would move on to the next one. That is, are there any known biomarkers or EEG, MEG signatures that could help tailor neuromodulation to individual patients' oscillatory deficits?
Yeah. That comes back to the question from Azuka earlier about who benefits from this and where does this work? And this is actually a crucial question. So, for example, we could theoretically use sharp wave ripples not just as the target of our stimulation, but also could use it as a so-called biomarker or outcome parameter to be used in a brain-computer interface. So, for example, it is already possible to use visually evoked steady-state potentials as the signal for a brain-computer interface. So there is nice experiments where we show one flickering stimulus on the left and one flickering stimulus on the right, and you can track attention of participants by just tracking the frequency of the visual steady-state evoked potential in the brain. So this works very, very well. So if we could do the same thing for sharp wave ripples, then we would have a biomarker of our high frequency stimulation in the brain. So far, we are mostly limited to neocortical responses because the deep brain signals originating from the hippocampus aren't really possible to pick them up with the EEG. Whether it's possible to pick them up with the MEG is still under debate. So there are some papers showing that you can record hippocampal sharp wave ripples with MEG, but this is still ongoing. And actually, we are ourselves working on a machine learning approach to detect sharp wave ripples from EEG signals. But I think it's still a bit premature to actually have this. So far, it's mostly neocortical responses.
Thank you. To add on to this question, wouldn't it be a possible approach to use something like the PET, so a time-delayed method, to indirectly measure the sharp wave ripples? So we stimulate, and then we give some substance with a, I don't know, 20-minute delay.
Yeah.
And so then we could guess, oh, this is at least the region where we would expect the sharp wave ripples to occur. And then say, okay, we think this is due to the activity that we would like to induce.
Yeah. That is an excellent idea. That is great idea. I will write it down. We haven't actually thought that much in this direction because our first question was still, can we actually induce the increased number of sharp wave ripples? And then the next question is, does this translate? So, but actually, PET, positron emission tomography is a great way of tracking this because this tracks neural activity for a longer time. So, for example, we could give the contrast dye, then do the stimulation so that we modulate the metabolism and then measure afterwards. It's actually a great idea. I haven't thought about this. Thanks.
Sure. It was just in my mind right now. So 2 questions left. This was one of mine actually, and we've also spoke about before. Have you thought of, or is there any known data about combining HBOT and non-invasive brain stimulation? I've also asked this already on the book this year, and nobody could tell me. But to explain, for those who don't know, HBOT is hyperbaric oxygen therapy. It is mostly done in stroke patients, and it has insane effects on different biomarkers like VEGF. There are studies that show a 300 to 900% increase in VEGF. So it is really a severe effect in regeneration. And for my opinion, it is very interesting to combine two such methods, because while being in this HBOT capsule, patients are in a very stable regenerative state where you also could add non-invasive stimulation, and maybe there's an interaction of those two effects. While there's better regeneration, they could also be better stimulated, maybe. But you've also said you don't know really about it, but a little bit about strokes in this context. So
yeah, I think this is again a super interesting idea, because I think let's assume that sensory stimulation can induce neural oscillations that can have not just direct cognitive effects, but also these effects on the, for example, synaptic strength or clearance of something. Then getting a patient into a state where we increase the malleability or the flexibility of the neural system would vastly improve these things. So a slightly similar idea is being used in the application of the amyloid beta antibodies, where a big block of the application of these drugs is the blood-brain barrier. So one way of increasing the efficacy of these drugs is temporarily disrupting the blood-brain barrier so that these substances can enter the brain easier. And there, TMS actually is one way of modulating the permeability of the blood-brain barrier so that substances can enter the brain easier. So if we could use any type of intervention to increase a state of increased flexibility in the brain or increased openness in the brain for any type of intervention, this would be super interesting. So one question there would be, are these patients awake in these states? So they are awake, lying in a tube, and then could be stimulated.
Depending on how much you're willing to pay, or how much the hospital was willing to pay, you can even sit in there.
Okay.
So prices depend on 7 to 10,000, the least expensive, to up to 350,000, where 5 people can sit around the table. It's basically just a capsule where divers also stay in.
Yeah.
2 atmospheres below, so in terms of pressure, 100% of oxygen. And then you're sitting there for 90 minutes, I guess, or 60 minutes, depends on if you are at the beginning or late on in your therapy. And so therefore you can use basic electronic devices, because there's just the delay of 2 atmospheres, and that's totally fine for most of our products.
That is a very cool idea. So the beauty about what we are working on right now is that we are developing a handheld device. So it's an augmented reality glass connected to a tablet that is delivering the stimulation. So it's actually very mobile. Let's talk about this.
I'm sorry, come on.
Let's talk about this.
Afterwards, you mean?
Yeah, well, at some point. I don't know what your future plans are.
I have quite a few, but definitely.
Okay.
So then, I was a little bit confused. I'm sorry. I wait too long, I think. But the last question that is recorded here, if it isn't already part of the talk, I would be interested to hear what is known about TVNS and its effect on memory formation.
Yeah. I quickly glanced over this. So let me quickly share my screen again. So the long story short is there is a very nice review on the effect of vagus nerve stimulation as memory enhancement. And yes, it works. So vagus nerve stimulation is, I quickly mentioned it. So you have the vagus nerve, that is one of the cranial nerves, and you can stimulate the vagus nerve non-invasively by stimulating, for example, in your brain, so in your ear. So you can have a little stimulator on your ear, stimulate the vagus nerve, and this has an ascending stimulus connection through the nucleus, through the locus coeruleus, and then on the thalamus, and then from there on it has brain-wide effects. So it's not an entrainment per se, but it's an activating of this vagus nerve pathway. There are some indications that through the pathway of the locus coeruleus, you can also activate the hippocampus, so you can activate these memory-related areas. This has some memory, so positive effects on memory. So for example, there's quite a few papers showing that in epilepsy patients or in older adults, you can significantly improve learning or memory in these patients. Unfortunately, as far as I know, no one has looked at the effect of the vagus nerve stimulation on the hippocampus. So for example, I don't know of any papers, for example, looking at sharp wave ripples and whether it's possible that this vagus nerve stimulation actually increases the number of sharp wave ripples. But again, this might be an interesting combination treatment where, for example, with the vagus nerve stimulation, we can get the hippocampus in a receptive state and then use a sensory stimulation to trigger these ripple oscillations in the hippocampus. So this is a very interesting side avenue. It's not related to entrainment, so that's why I didn't go into a lot of detail. But yes, vagus nerve stimulation can improve memory.
Okay, then thank you very much for answering all of our questions, very, very detailed, and taking your time. I think at this point there aren't any more questions in the chat, so I would say thanks for everybody who's still here. And yeah, thanks for joining this second Neuro Talk evening. Thank you, Dr. Keil. And
Thank you for having me.
And then have a very nice evening. We might wonder why there's another post edit. I'm terribly sorry. I've got too much involved in the discussion with Dr. Keil. So therefore I forgot the ending slides, but there's only a little bit more information. So as mentioned, there is the Neuro Talk evening with Professor Julian Lee about cognitive neuroscience of aging on 28th of August. But we also scheduled one Neuro Talk evening before, but this will announced within the next few days. So maybe look out for it because we don't have a title right now, so we can't really announce anything because there's only a topic and this is a very broad topic. So therefore, we can't do a specific announcement, but we will. Also, if you would like to hear more of Dr. Keil, you can watch his YouTube channel, you can visit his website, and maybe also his LinkedIn. This is included in one of the first slides. And also, if you would like to join us, the network, maybe our leads team, just feel free to message us. Join our Discord, our WhatsApp group. We're happy about everybody who's there and showing some interest in neuroscience, regardless of from which department you are. Might be informatics, psychology, biology. We're constantly searching for new perspectives, and yeah, we're really happy to see anybody coming new to our network. And so therefore, thank you for joining the Neuro Talk evening, or having joined the Neuro Talk evening. Have a wonderful evening, and yeah, see you next time.